Assalamu alaikum and welcome everyone to the second part of molecular biology ASCP preparation series, Molecular Techniques. I'm Dr. Lai Abdulati and I'm really excited to share my knowledge and experiences with all of you through this series. So let's dive in together. Today we'll continue in one of the most essential tools in molecular biology, third generation sequencing, specifically Oxford Nanopore sequencing and Pacific Biosciences SMRT sequencing. So, welcome to the season.
Break it down small like DNA. Learning the code where secrets stay. Chromosomes dance under the light. Molecular dreams will take their flight. Molecular magic, it's in your hands. Unlock the science and take a stand. Study hard, let your passions ignite. ASCP success is within your sight.
Alright, third generation sequencing represents a major advancement in DNA and RNA analysis, addressing the limitation of previous technologies, unlike first and second generation sequencing, which require DNA amplification. Third generation sequencing enables the direct sequencing of single molecule, making it faster, more accurate, and capable of handling complex genetic structure. Key features that make third generation sequencing stand out. First, long read sequencing. It means reads can extend beyond 100 kilobases, which help resolve difficult genomic regions like repeats and structural variants. Second, real-time sequencing. Data is generated as the DNA is being sequenced, speeding up workflow and improve accuracy. Third, single molecule analysis. No amplification means fewer error and more faithful representation of original genetic material. Lastly, epigenetic detection. Third generation sequencing can directly identify modification like DNA methylation, offering insights into gene regulation and disease mechanism. The leading technologies in the third generation sequencing include: number one, Pacific Bioscience SMRT sequencing uses zero-mode waveguides to track DNA polymerase activity in real time, so generating highly accurate long-read sequences. Second one, Oxford Nanopore sequencing uses protein nanopore to pass DNA through the electrical sensor, enabling ultra-long reads and real-time analysis. Now, let's move to the question section and we'll start with the Oxford Nanopore sequencing. Alright, the first question we have today: which sequencing methods uses a nanopores to detect changes in ionic current? The correct answer is Oxford Nanopore sequencing. Actually, Oxford Nanopore sequencing works by passing a single strand of DNA or RNA through a nanopore and detecting changes in electrical current. This current changes corresponding to the different nucleotide, allowing base by base identification. Alright, let's move on. In Oxford Nanopore sequencing, what is typically used to control the speed of DNA passing through the nanopore? The correct answer is motor protein. Actually, a motor protein is attached to the DNA strand and regulates its movement through the nanopore. Alright? This ensures that the DNA passes through slowly enough for the sequencer to detect and distinguish each nucleotide accurately. Alright, let's move on. Alright, the next question here. Which feature is the major advantage of Oxford Nanopore sequencing compared to the other platforms? Alright, the correct answer is ultra long read lengths. As we know, Oxford Nanopore sequencing is especially known for producing ultra long reads, sometimes exceeding hundreds of kilobases. This is highly valuable for resolving repetitive regions and complex genome structures. Alright, let's move on. Okay, next question here. In Oxford Nanopore sequencing, what is directly measured to determine the DNA or RNA sequence? The correct answer is electrical current change. Alright, as we know, Oxford Nanopore sequencing detect small changes in electrical current as nucleotide pass through the nanopore. So each base causes a distinct disruption in the current, allowing the device to infer the sequence. Alright, let's move on. Okay, this question here. Which sequencing technology can directly sequence RNA molecules without conversion to complementary DNA? The correct answer is Oxford nanopore sequencing. Actually, Oxford nanopore sequencing or technology is a unique because it can directly sequence RNA molecules by passing them through the nanopore. All right. This approach eliminates the need for complementary DNA sensitivity step, preserving the native RNA information. All right. Let's move on. Okay. In Oxford nanopore sequencing, what is the role of the nanopore embedded in the membrane? The correct answer it acts as a tensor for nucleotide detection. As we know, these nanopores act like a molecular sensors. As the DNA or RNA strand move through the pore, it disrupts the ionic current across the membrane. And these disruptions are analyzed to determine the sequence. Okay, let's move on. For this question here, which sample type can Oxford nanopore sequencing handle directly without extensive processing? One of X4 nanopore strengths is the ability to sequence native DNA or RNA directly without requiring amplification or chemical labeling, preserving epigenetic modification like methylation. Alright, let's move on. Okay, this question here. What is the unique capability of X4 nanopore sequencing regarding epigenetic analysis? The correct answer, it can detect DNA methylation during sequencing. Why? Because modified bases cause distinct changes in electrical current signature. All right, let's move on. Please, if you found this content helpful, don't forget to like, share, and subscribe. Your support means the world to me and help me create more valuable content for you. Thank you for being part of this journey. Let's continue. All right, now we'll move to the second part of third generation sequencing, Pacific Bioscience single molecule real-time sequencing. All right, which sequencing platform is known for implementing single molecule real-time sequencing? The correct answer is Pacific Biosciences developed smart sequencing which observe DNA synthesis at the single molecule level in real time using fluorescently labeled nucleotides. All right? So the first question we have here, what does SMRT stand for? SMRT stands for single molecule real-time sequencing. This technology allows observation of DNA synthesis by a single DNA polymerase molecule in real time without the need for amplification. All right? All right. In single molecule sequencing, what does the term real-time sequencing refer to? The correct answer is base detection occurs immediately as the polymerase incorporates nucleotides. All right, let's move on. All right, what type of nucleotides are used in smart sequencing? The correct answer is fluorescent labeled nucleotides. So each of the four DNA bases is attached to a unique fluorescent label, enabling real-time detection of base incorporation events. All right, let's move on. Okay, this question here. What is the primary enzyme used in smart sequencing to incorporate nucleotides? Smart sequencing uses a highly processed DNA polymerase that can incorporate fluorescently labeled nucleotides during real-time observation of DNA synthesis. Okay, in smart sequencing, what is the name of the small reaction chamber where sequencing occurs? All right, sequencing reactions take place inside zero-mode waveguides, which are tiny holes that allow observation of individual DNA polymerases working on a single molecule of DNA. Okay, next question here. What is the key feature of the zero-mode waveguide that enables smart sequencing? The correct answer, it limits the observation volume. It means that the zero mode waveguides confines the observation volume to a tiny space, allowing the detection of fluorescent signals from a single DNA polymerase at work. Let's move on. All right. Which type of DNA molecule is typically prepared for smart sequencing? The correct answer is closed circular DNA. Actually, smart sequencing or SMRT sequencing relies on a special type of DNA molecules called smart bell templates, okay, which are structured as the closed circular DNA. This circular design is essential because it allows the sequence process to continuously read the DNA without interruption, okay? All right, let's fix the information with this question. The question is, what is the smart bell in smart sequencing? Smart bell is a DNA library construct, as I said, where hairpin adapters are ligated to the both end of DNA fragments, all right, creating closed circular that allows multiple passes over the same DNA during sequencing. So the correct answer here is circularized DNA template. Okay, which feature allows smart sequencing to detect epigenetic modification like methylation? The correct answer is real-time polymerase kinetic. As we know, changes in polymerase kinetic, such as the speed and post duration when encountering modified bases, enable smart sequencing to detect epigenetic modification without chemical treatment. All right, let's move on. All right, in Pacific Biosciences sequencing, what helps in sequencing highly repetitive genomic regions? The correct answer is long read lengths. Actually, long reads generated by smart sequencing are particularly useful for spanning and accurately assembling repetitive regions of genome that short reads often cannot resolve. So, by using long read smart sequencing, scientists can get a more complete picture of organism genome without the limitation of short read technologies. All right, let's move on. Okay, what is the main characteristic of single molecule sequencing technologies? Correct answer is sequences individual DNA molecule without amplification. All right, let's move on. Okay, next question here. Which of the following is the major advantage of single molecule sequencing over traditional sequencing methods? The correct answer avoid BCR amplification PS. Actually, by directly reading single molecule sequencing eliminate amplification PS, allowing for more accurate representation of difficult or low abundance sequences. All right, let's move on. Okay, next question here. Which type of sequencing can best detect long range structural variants and large genomic rearrangements? The correct answer is single molecule long read sequencing. As you know, single molecule sequencing platform like Pacific Biosciences and Oxford Nanopore provide long reads that span complex genomic region, all right, making them ideal for structural variance detection and genome assembly. All right, let's move on. Okay, next question here. What is the practical application of single molecule sequencing in clinical research? The correct answer is resolving complex genomic regions in rare disease diagnostics. As we know, single molecule sequencing technologies are valuable in clinical genomics, especially for resolving repetitive or highly variable regions involved rare genetic disease that are challenging to study using short read platforms. All right? All right, next question here. What feature helps smart sequencing identify full length RNA transcripts? The correct answer is long continuous reads. As we know, long continuous reads allow smart sequencing to capture entire RNA transcripts without fragmentation, which is important for transcriptome studies. All right, let's move on. Okay, next question here. Which major challenge does single molecule sequencing help overcome in microbial genomics? Because single molecule sequencing provide long reads, it is excellent for assembling complete microbial genome, including difficult to sequence plasmids and repetitive regions, which are the crucial in studying antibiotic resistance. All right, let's move on. All right, next question here. What makes single molecule sequencing ideal for full length isoform sequencing? All right. Actually, single molecule sequencing technologies, particularly such as the Pacific Biosciences and Oxford Nanopore, can read full-length RNA transcripts directly, all right, or after complementary DNA conversion. So, it's making them powerful for studying isoform diversity and gene expression. Okay, so the correct answer here is ability to sequence entire transcript without fragmentation. All right, that's all for today. I hope you enjoyed this lecture. I look forward to hearing from you. What you think of it? Please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. And don't forget to click to watch the previous videos. Salam.
Break it down small like DNA. Learning the code where secrets stay. Chromosomes dance under the light. Molecular dreams will take their flight. Molecular magic, it's in your hands. Unlock the science and take a stand. Study hard, let your passions ignite. ASCP success is within your sight.